Good morning, class. So today we will continue what we had started yesterday, that is neural control and coordination. And yesterday we have talked about the what a threshold stimulus is, how an impulse is generated and conducted along the length of the axon, and then when an axon makes a synapse with another neuron, then how the impulse is transferred, right? And then we are going to take up the central nervous system, which just the beginning part I have taken up with you, the parts of the brain I had mentioned. So now we can talk about the parts of the brain, starting with forebrain, of course, or rather few things we can first mention and then I'll draw a diagram. Okay, forebrain. Now forebrain, as I told you, I had taken three regions there. Number one, olfactory lobes. Olfactory lobes. Now olfactory lobes, these are one pair solid lobes. So you see, wherever in whichever part of the brain we mention solid, so that means there isn't any ventricle in it. So it is without any ventricle, and so that's what we mean by solid. And this one receives impulses, sensory impulses, that is, receives sensory impulses from olfactory epithelium. I hope everybody remembers olfactory epithelium is on the roof of the nasal cavity. So there, from there the impulses are reaching here from the olfactory epithelium. Okay, so that is just this much you need to know. We have this one very small, but if you see the brain of the shark, their olfactory lobes are huge, quite big, because they have very keen sense of smell. Okay, then next one, that is cerebrum. So in the forebrain, next part, that is cerebrum. Now this cerebrum, it is the largest part of our brain, and this one comprises two cerebral hemispheres. Two cerebral hemispheres. So if you see the brain from the top, you will find there is a longitudinal deep invagination, that is sulcus basically. So longitudinal sulcus is there, which is dividing the cerebrum into two hemispherical parts, so the right and the left hemisphere. Okay, and two cerebral hemispheres which are connected by corpus callosum. Corpus callosum. Now corpus callosum is a thick band of nerves, white tracts rather, because inside the CNS we won't use the term nerve, rather tract. So this is a thick band of white tracts and it is connecting the two hemispheres. Okay, so these are tracts of white fibers, and this is a very important, you can say, a kind of route through which the left and the right cerebral hemispheres, they are passing on information from one to the other, right? And this corpus callosum, another thing that you need to remember is that this corpus callosum is present only in the mammalian brain. Okay, so corpus callosum, one another thing we can add on, this is present only in mammals. Present only in the mammals. Okay. Now you see the entire each of the cerebral hemisphere, you will find that it has highly convoluted surface. Okay, so each cerebral hemisphere has outer convoluted surface, which appears gray, and this is called the cerebral cortex. Okay, so this is called the cerebral cortex. So this is the outer one. outer area, that is the cerebral cortex, and this outer area is highly convoluted and it is grey in color. So when we mention grey in color, that means what must be here? Non-myelinated and structures. And what are non-myelinated structures here? The cytons. So it is having groups, a large number of cytons here which are clustered in this area. Okay, so this has large number of cytons. Okay, now the convolutions which are here, those convolutions are the gyrus and the sulcus, you know that. Okay, so these folds, if you see these folds or convolutions as we mentioned, so the outer one, which is like which are the ridges which are coming outward, so the ridges are the ones which are called the gyrus, or the plural would be gyri, and the furrows, that means which are going inside, those are the one which are called sulci. So the entire surface is like that. Now if we just take a diagram, so I can mark certain areas on this cerebral cortex. So you see, if you see the brain from the lateral side, from one side if you are looking at it, it more or less appears like a punching glove, right? That kind of shape it gives. So it's very easy to draw. You can see, you can draw a hemispherical structure like this. So this is how the hemisphere is, and then this is how it goes. So this would how the one hemisphere will appear, the other hemisphere is on the other side of it. And since we have drawn, let us draw the other parts as well, and like this. Okay, now in this, the entire surface, as I told you, you won't be seeing it uniform surface, rather number of folds are there. So this more or less, but there are some folds which are going along a longer length, and on that basis we divide each cerebral cortex into 4 lobes. Okay, so there is one here, another one, another sulcus here like this, and this one of course is also sulcus. So in this case, the names of these lobes are on the basis of the bone which is in front of them. Like here, you know, it is frontal, then parietal, occipital at the back, and here it is the temporal. So the same naming we use. So here let me first mention one thing, that each cerebral cortex, that means for each cerebral hemisphere, each cerebral cortex is divided into 4 lobes. Okay, so each cerebral cortex is divided into 4 lobes. Okay, so let us mark these lobes. Here this is frontal lobe. Then here this sulcus, this is called the central sulcus. Central sulcus. And this one is the parietal lobe. Parietal lobe. Then this one is parietal, or parieto-occipital sulcus, and this one is the occipital lobe. Okay, and here this one is the temporal, you see. This is temporal lobe, and temporal lobe from the parietal, you see this part here, this is which is separating it, that is called the lateral sulcus, or the fissure, Sylvian fissure also it is called, lateral sulcus or Sylvian fissure. Fissure and sulcus are more or less similar things, depressions, right? And this part here is the pons, you see, that is hindbrain. The midbrain and the diencephalon, the thalamus, hypothalamus, we won't be able to see because the cortex is literally wrapping it, okay? So that's why we won't be able to see it until unless we take a section through it. And pons, this is medulla, and this is the cerebellum. And in this section, when you take a section through the cerebellum, you will see a tree-like pattern over here, and that's what is arbor vitae. Arbor vitae, that is tree of life. That literally means tree of life. Okay, now you see in this part there are certain specific areas, not all of course, because there are nearly 44-45 areas, so that is beyond the scope of the 12th syllabus, but some important areas we can certainly mark. So you see frontal lobe is mainly concerned with sending motor commands, voluntary motor commands. So whenever you are moving or you are writing, that means when you are thinking and then doing, so that command is coming from your frontal lobe. So this one is responsible for the motor commands, and in this we can mention from here motor commands are sent, and in this one specific area certainly I would like to mark that particular area on the cortex. This is called the Broca's area. I'll just rub this side. I think this side everybody has already done, so I'll have a little more space. So this one is Broca's area. Now Broca's area is your motor speech area. That means when you speak, all these muscles that we will be using, that all commands come from the Broca's area. Okay, so this is your motor speech area. Okay, area already is written. Now parietal lobe is receiving all the various sensory impulses. So this one receives sensory impulses from various parts of the body. You see impulses like touch, pain, temperature difference, you touch a hot object or a cold object. So all those sensory impulses will be reaching over here. Okay. Then here occipital, once again, that is, this one is special. Here all the visual impulses reach. Okay. So when image is formed on the retina, that impulses will be reaching to the occipital lobe where it will be interpreted. Okay, so this one here is having visual area. This part is yes. And in the temporal, I'll just mark few areas. I'll write numbers rather. Okay, so here we have, let me put 1, another 1, 2, like this. So this is auditory area. That means whatever we hear, that sound will be coming here finally. Okay, and that will be interpreted. So this is the auditory area. Then there is olfactory area. So just now I mentioned the olfactory lobe will be receiving the impulses, but after synapse, the final integration will be done at the olfactory area, which is in the temporal. Okay, so olfactory area is there. And one more area let me mark, that is in the parietal, and it continues in the temporal also. This one, let us mark it 3. This one is Wernicke's area. Wernicke's area. Now Wernicke's area, you see, is your sensory area, which is for language. And language, when you think about it, is not only the spoken words that you are able to understand, but the written language. When it is written, of course you know that language, then only you will be able to interpret what all is written there. So this comprehending the language, whether it is written or spoken, that would be possible because of Wernicke's area. Okay, so this one comprehends written and spoken language. Okay, and these areas, you see, they are all interconnected also. Like this Wernicke's area and Broca's area, they are there are tracks which are connecting them. Like when I pose a question to you, you will be hearing and understanding. You will be hearing it, of course, that interpretation would be from the auditory area. You will understand it because of the Wernicke's area, and then you will, if audio was available, you will speak out the answer. That would be because of the Broca's area, right? But here you won't be speaking out. That's not allowed in this class. rather you will be typing in your answer. So that means that's going to be a motor command. That will be, once again, you will be using your frontal lobe. So you see all interconnections are there. So one type of area is interconnecting with the other, and that is the reason that our brain has so enlarged. There are many areas which integrate this sensory and motor commands, and those are called the association areas. So maximum, like suppose here I have made a visual area. Now this, around this visual area there would be some area here which is called the visual association area. Same way around the auditory area, everywhere that the maximum part of our cerebral cortex is actually contributed by the association areas. So we can just mention this one also, association or association cortex also we can say, areas or cortex, that's one and the same thing, association cortex. So this one integrates sensory informations and helps in intersensory perceptions. So different sensory informations are going to be perceived, intersensory perceptions, and that helps in developing our memory. So that's why while studying also, usually you see the educationists will say that you should apply all your senses. It is not just that you are taking one way, that you are just listening, or maybe you are just viewing it, like presently you are viewing the lectures, but along with it, when you are writing it down also, and then you see to it again, so that means you are using different parts of your brain, and as a result that will help in developing better memories. So as many senses we can involve together, that helps. So anyway, this was about the cortical part, not much, not a lot of detail, but as much were required I have given you here. Now coming to, let's cut it, okay, because the cortical area is very slight, you see, just around a centimeter or so, but within it then there are so many neurons. We have billions of neurons here, and you can imagine the complexity of our brain. Now when we cut it, so what we will see underneath, there is a white region which is called the cerebral medulla. Okay, so next one, let us mention so let me see, okay, cerebral medulla. Now this one is made up of white matter. Now when we say white matter, that means it is having white tracts. So you can imagine something like this, like suppose if this is the cyton I am representing. So in the cortex area, the cytons one after the other, they are arranged, and their axons will be running deeper, and then another layer of cytons, right? So this is just the tip of the iceberg. So we can imagine this part is the gray area part, which is the cortex part, and here you see all the axons here, this is the white part, okay. So this is made up of white matter, and in this what will be present? Myelinated axons, myelinated tracts are present. So bundles of axons, you know, that is tract. So white matter, and this one consists of myelinated tracts, myelinated tracts. And it is not just white myelinated tract, because we have just now mentioned that this cortex is highly convoluted. So you see this entire cortical layer, if it is forming a sulcus, so that means the gray matter is going, or it is penetrating into the white matter, and those areas in the white matter where these cell bodies, the clusters of cell bodies can be seen, those are of course what? Nuclei. Okay, so myelinated tracts, and also nuclei, which are of course gray matter of cortex can be seen. Okay, and in this there are some important nuclei which we will be mentioning presently by taking up the limbic system. Then another part you see is, now what is underneath then, like suppose this is the cortex, this is the medulla, but then here there is a cavity inside. So you can try to imagine that inside there is going to be a cavity, and this cavity is filled with the cerebrospinal fluid, and that is the ventricle. Okay, so here lateral ventricles or first and second ventricle they are also called, because in our brain these are the first and second, because olfactory lobes you see they are solid. So first and second ventricles, so these are present in cerebral hemisphere. So, and it is no the books clearly write that it is not that the right cerebral hemisphere, the ventricle which is present, you have to take first, or for left you have to take first. They are simply called first and second. Okay, so these are present in the cerebral hemisphere, present in the cerebral hemispheres, these lateral ventricles, and these are of course filled with CSF, and these. Okay, so this will do about the cerebrum. Then coming to the third part of the forebrain that we mentioned, and the third part of the forebrain is the diencephalon. So diencephalon, this is a very small part, less than 2% of the brain, but a very important part you see. So in this diencephalon there is, in this also there is a ventricle. There is a ventricle in the diencephalon that is called now the third ventricle. Okay, so this one has third ventricle, or which can also be called or diocele, and all these ventricles you see they are connected. So it is connected to first and second ventricle, it is connected to first and second ventricle by and small aperture which is called foramen of Monro. Foramen of Monro. Okay, now in this diencephalon there is basically it is three parts: epithalamus, thalamus and hypothalamus, but epithalamus is the roof part which is non-nervous, so we can skip it and we can directly move on to thalamus. Okay, so thalamus, once again you see it's a nuclei, so it's a pair of solid nuclei, very small part but very important, because its function is, it's called the relay center, because whatever sensory informations you see we are reaching, they are reaching to the brain, they do not directly go to the brain, they would be first reaching to the thalamus where the neurons which are present, so nuclei you see, so there are number of cytons, it will synapse over there, and from there it will send to the proper center where it should be sent, right? So it is like a router that we have for Wi-Fi. So same way it is receiving all the sensory informations and then routing it to the exact center of the cerebral cortex. Okay, so this one is the relay center where sensory impulses are received and sent to cerebral cortex areas. Okay, so that is it. Then the other one, now I think we can rub this off, everybody must have seen the diagram. Okay, so next one, hypothalamus. So hypothalamus, this is the one which is the floor of the diencephalon. And this hypothalamus, you see, it is connected to the pituitary by a narrow stalk, connected to pituitary by a stalk called infundibulum. Okay, called the infundibulum. And this hypothalamus has number of important centers. And you can say it is also a link between our nervous and the endocrine system. Yesterday I told you that nervous and endocrine system, both of them are concerned with control and coordination of our various organ systems. And so these two systems are then, they are of course connected through hypothalamus. So it's a neuroendocrine link, and it has centers for, it has number of centers like for temperature regulation, thermoregulatory center. And this thermoregulatory center, once again you see, it will be present only in case of warm-blooded animals. So in a cold-blooded animal, the hypothalamus will not have any thermoregulatory center. So if you are studying the brain of a frog or a fish or maybe a reptile, then there won't be this thermoregulation part. Okay, so it is only in case of birds and mammals that you will have this part in the hypothalamus. Then it has for appetite, that is feeling hunger, appetite. Then there is, and not only appetite, hunger center, but satiety center also. Like once a person starts eating, after certain time, because as the food digests, our blood glucose level rises, and the sensor here in the hypothalamus is able to sense it. And so further you will feel that now you are full, you cannot eat anymore. So if somebody's satiety center is removed, that person will keep on eating. They won't be able to stop. They will never know that when to stop. Okay, so that is satiety center. Then also there is thirst center here. These all centers you see, they are very much in the hypothalamus. Then apart from that, hypothalamus has neurosecretory cells. Neurosecretory cells secrete hormones. So there are a number of hormones associated with hypothalamus. We'll be doing a long list when we take up the chemical coordination chapter. So this was about diencephalon. Now one more thing we can mention with diencephalon, because since I skipped epithalamus, on the roof part of the diencephalon there is another endocrine gland, that one is pineal. So that at least we can mention separately, pineal gland. This is on the roof of diencephalon, or roof of third ventricle also it can be written as. Okay, and pineal gland, of course you know, it is our biological clock. So our with the change in the day and night, the sleep wake cycles and the metabolism, our basal metabolic rate doesn't remain constant all through the 24 hours. We are basically diurnal animals, but with the advent of light we have tried to become nocturnal, keeping awake till late night, but actually we are diurnal, so our entire system works that way. So our metabolism is quite high in the morning, and as we progress through the day towards evening, it keeps on decreasing. So that kind of circadian rhythms are also maintained by pineal secretions. Okay, so it is, so that's why it is called biological clock of the body. Okay, so this is there. Just a sec, this AC is making a lot of noise today. Nothing, I'll have to switch it off only. Okay, now so this completes with the forebrain part. Forebrain was the most extensive part also, but rest of them, they are very short and usual questions. And once again, let me tell you, as far as previous year question papers of NEET exam we see, there are more questions on the nerve impulse, synapse, but fewer questions from the brain actually, right? And then once again, receptors, eye, ear, those are very important. A lot of questions you get from that. So don't do too many details about the brain. At least be thorough with the NCERT, and few extra things that I am telling you, that much will serve the purpose. Okay, so next thing, that is midbrain. Midbrain. Now within the midbrain, there isn't any ventricle you see, but there is a narrow canal which is filled with CSF. So this one has cerebral aqueduct. This is what the NCERT writes. We can write the full form also. Aqueduct of Sylvius, or which is also called iter. So this is a narrow canal filled with CSF. A narrow canal filled with CSF. Okay, and this one connects the ventricles. Now you see, if you see the brain of any lower organism, let us say fish or frog, all these you will find one after the other very nicely placed, easy to study. Like you will have the olfactory lobes, and then the cerebrum, and then the diencephalon, and then the midbrain part would be there, and then the hindbrain part would be there, one after the other. Okay, but in our case what has happened, the cerebrum has increased so much so that it has literally enclosed our diencephalon and the midbrain. So that's why we are not able to see. Yes, Shubham, pineal is sensitive to light. Exactly, it is because some of the nerves which are coming from the retina, their branches are going into the pineal also. So certainly it is sensitive to light. Okay, so in that case, you will see that here this is the third ventricle in the diencephalon, fourth ventricle we will see in the hindbrain. So this aqueduct of Sylvius is connecting what? The third and the fourth ventricle. So that once again is a question. So this one we can mention, it connects third and fourth ventricles. Then in this midbrain there is the structure that we had mentioned, that was corpora quadrigemina. Corpora quadrigemina. Now these are four solid lobes on dorsal side of midbrain, and these four solid lobes, you see, they are now this name NCERT hasn't given, but anyway we'll take it. So let us just let me just give you a simplified form. Like these are four, two lobes here, and the upper one, that is called the superior colliculus. Plural would be of course colliculi, two superior colliculi, and the lower ones, these are inferior colliculi, or singular will be colliculus. So these four lobes are there. Now these, the upper one of course, this is receiving some of the branches from once again the retina. So you see from the retina, the optic nerve which is coming, some branches will be coming like this, and from the left to the right, crisscross, this is how it goes. So where the two optic nerves are crossing, that part is called the optic chiasma. So chiasma is the X-shaped structure that you get, that is chiasma, and this is of course branch of optic nerve. So you see, that means this is also getting the sensory impulse. Like when you see something, you try to, if you want to really see it nicely, you will turn your head, direct your gaze towards it. That's what is called the visual reflex. So superior colliculus is helping you to for visual reflexes, while when you hear a sound and you turn your head so that you move towards the sound or maybe away from the sound, that is controlled by the inferior colliculus. So this one, superior colliculus, this helps in visual reflexes, and this one helps in auditory reflexes. Okay. So this was corpora quadrigemina part, solid part. And once again, corpora quadrigemina is also something which is present only in case of mammals. Now prior to mammals, other vertebrates, if you will see, that is from fishes to birds, in the midbrain there are only two lobes, just two lobed structure, and that is called optic lobes. But as far as mammals are concerned, optic lobes is not the term which you will be using. You will be using this term here. Okay, and then next one, that is the crura cerebri that I told you, which is also called cerebral peduncles or crura cerebri. Okay. Now in this, these are white tracts once again. So these are white tract, that means myelinated fibers, white tracts that connect. Now since midbrain is in the center, so forebrain and hindbrain that connect fore and hindbrain. Okay. So you see the midbrain, from here also some of the motor commands are sent, like you see when we are moving our eyeball, that all comes under part of the visual reflexes. So the muscles to move the eyeball, they are also very much a skeletal muscles. So for that the command is going, that would be a motor command. So this one does send some of the motor commands from this area. Okay, and then next one, the last part, that is hindbrain. Now in the hindbrain, let us first begin with cerebellum. Three parts I had mentioned yesterday: cerebellum, pons and the medulla. So first starting with cerebellum. Cerebellum word comes from, literally it is little cerebrum. So it is very similar to that in the form that it is also highly convoluted surface it has, but it is not hollow. It doesn't have any ventricle. It is solid. Okay. So this one is solid structure having outer convoluted cerebellar cortex of grey matter, here also the same way, of grey matter. Okay, and here also sulcus and gyrus would be present. Startle reflex, Shivani, would be a reflex, no? So some, of course, brain, because this is one common error, because in class 10th students are taught that reflex actions are controlled only by the spinal cord, brain is not involved. But of course there is impulses, they do go to the brain also, and when we are born, nearly more than 200 reflexes are already wired into our brain. So this is also very much inborn reflex, and this is this will be of course from the once again from the midbrain part only. Okay, so outer convoluted part, and then the inner is cerebellar medulla, which is of white matter. And here you had seen, here also because of the sulcus, the grey matter is penetrating into the white matter, so that the white matter appears tree-like, which I had drawn in the diagram called arbor vitae. So that was the structural part. That is now what does the cerebellum do? Cerebellum is for maintaining balance and posture. Okay, so this one maintains balance and posture. Then you see, we mentioned that the frontal lobe of the cerebral cortex, it gives motor commands. So the motor commands which are sent by the frontal lobe, they are quite crude, but cerebellum and cerebellum also are connected, you see. So cerebellum, it makes it more refined, more precise. Like you see, when small young children are there, because number of brain cells are not going to increase. You know that nerve cells do not divide. But those, they have very gross movements. If they have to pick up anything, they'll just put their entire hand and pick it. They can't pinpoint something or take hold of it. So that is because initially when a child is born, all these synapses between the various areas have not yet developed. So as they are made to practice more and more, so they will synapses will develop, and then the process becomes more refined, and that refinement comes from this. Okay, so here we can just mention it refines the voluntary movements initiated by cerebrum. And you see, when a person is under alcoholic effect, So their movements, they become quite gross. And if a person is to be, like even police men use it, even if they don't have that breath control analyzer with them, they can easily find out that the person has taken alcohol or not. So they can just ask the person to get down from their car and walk in a straight line. They won't be able to walk. And all the more simple, even if the person is not ready to get down, they can just ask the person to put their fingertip on the nose tip, and the person who is drunk won't be able to do it. So that means this is not working, the refinement is not there, and accordingly the person is easily caught. So anyway, this was about cerebellum. Then coming to next part, that is pons veroli. As you know, the word pons itself means bridge. So this is also having number of tracts which are connecting various parts of the brain. Okay, so has tracts which connect different parts of brain, cerebrum to cerebellum, so on, and even cerebellum has lobes, so connecting the lobes of that. Okay, and then if you haven't forgotten, in the respiratory system we mentioned there is one center here related to breathing, that is pneumotaxic center, that is present here in the pons. So this one is having pneumotaxic center. Okay, and then the last part, that is medulla oblongata. Medulla oblongata. Now medulla oblongata is the part where there is a hollow space within, and this one has fourth ventricle in it. Okay, there is fourth ventricle that is there, and then in this, just a little about this, on the roof of this fourth ventricle there are three apertures, you see. So on the roof of fourth ventricle there are three apertures. Okay, three apertures, and these three apertures are names are, I don't think it's very important, but anyway, one is lateral in position, slightly lateral paired foramina. Foramina would be plural for foramen, foramina luschka. Okay, and the other one would be other one is the single median single foramen magendi. Okay, now from these apertures what is happening, the CSF from the fourth ventricle passes into the subarachnoid space. Okay, so CSF moves from fourth ventricle to subarachnoid space, and subarachnoid space has venous channels where it will be going into the venous blood. So this way you see the CSF circulates. Then this was one part. Then in medulla you see there is reversal of the grey and the white matter. Now the white matter becomes external and grey matter is internal. Okay, so in medulla grey matter is internalized, and the same thing will continue in the spinal cord also, you see. So now the grey matter is internal and white matter is external. Now, and in this medulla number of involuntary, number of involuntary controls are associated with medulla. Involuntary controls are associated, like you see, you have studied right from digestion. If you start, the deglutition center, vomiting center, right? So deglutition, that was swallowing, vomiting, then coughing, sneezing, respiratory rhythm center, cardiac center, and also you see vasomotor center, that is which will control that when due to blood pressure which blood vessel should means the blood vessel should dilate so as to bring down blood pressure. So that is the vasomotor center, so on. So many you see, that's why I told you the brainstem part, if it is damaged, the person cannot survive. So this was, and this medulla of course joins with the spinal cord, and that is coming, it is passing out through their aperture in the occipital bone, remember, the foramen magnum. Now one more thing before we end with the brain, that is limbic system. So that I am taking separately. Okay, so I think this much part must be over by this time. I'll rub half the board. Okay. So here you see, coming to this limbic system. Now this limbic system, you see, this is our emotional brain. Emotional brain. Now in this case, what is making up the limbic system? There are a number of nuclei, you see. Nuclei, I told you, these are group of cytons. And in this even hypothalamus is included. So that means basically it is in the forebrain where the hypothalamus, and around it there are certain nuclei which are all interconnected by tracts that is present, and that is taking up the shape of a lip. That's why it gets the name as limbic system. Okay, so this one is in forebrain, number one. Number two, this one comprises number of nuclei arranged around hypothalamus. So this is quite a complex system, you see, but we'll mention just few nuclei in it. So one important nuclei here in is amygdala. Amygdala, you see, is in the shape of an almond, because these nuclei are of different shapes, because when the sulcus is going in, so it can take different shapes as it is compressed, right? So here the shape is that of an almond. So this is paired almond shaped nuclei. Now what the amygdala does, it is because of the amygdala that we have the emotion of fear, the first thing. Okay, so, and that is also important, because if amygdala, normally it is so deep seated in accidents and all, it doesn't easily get damaged, but it was deliberately done in monkeys, and it was seen that even if a snake was put before them, they lifted the snake, while in the wild the monkey will quickly run off or climb a tree if there is a snake. So you see, fear also is a very important emotion, because that helps the person to protect himself or herself. Okay, so this is responsible for emotion of fear. But sometimes with fear, when it is not, one is not getting anywhere, then that fear turns into anger, rage. Okay, so fear and rage, extreme fear leads to rage at most of the time, because if a person suppose is running and another person is trying to kill that, and there is a dead end, they cannot run anywhere, then of course that person will try to hit the attacker, because when the person thinks that there is no other way out, right? The same thing here happens here. So anyway, this is one, amygdala. The other one is hippocampus, and hippocampus, if you remember, hippocampus is the name of seahorse, the scientific name of seahorse. So this one is a seahorse shaped nuclei, once again paired. Okay, so paired seahorse shaped nuclei, and this one is responsible for converting short term memories into long term memories. So this one converts short-term memories into long-term memories. And with short-term is like when you are listening just now to the class, you will remember these things for the next 15-20 minutes. But if you don't revise it again slow, because normally it happens when there is too much influx of information, which is nowadays happening. So most of the things we don't remember. We don't give the brain any chance to integrate it. So that is the reason that we should give a little chance, and once we hear something, we'll remember it for few minutes, and then by the time the nerve impulses will be going to other parts of the brain, where then it is going to be stored. And once again, you will say, what is memory? Means it's abstract. It's because memory is something so abstract. But if we go into the biochemistry of it or the molecular biology part of it, so there too the nerve cells, when they are stimulated, so in some of the cells some special kind of protein synthesis will take place, and the memory once again is stored in that manner. So that is much detail we'll go into, and there is no particular part of the brain that you will say that here all the memories is stored, not at all, because we have different memories. We have a memory of a smell, like you had a very nice perfume with you and you remember the smell. There is memory of something what you see, and there is memory of something you hear. As soon as you hear the tune, you immediately are able to recognize which song number is it. So there are so many different types of memory. Then the way how we wear our clothes, there is particular type to drape it and all that. That is also memory. That is procedural memory, how we work. And so you cannot say that there is only one part of the brain which stores the memory, right? So I think I've made it clear. So these are the two main ones, and as far as NEET exam is concerned, I think these two will be sufficient. Otherwise, once you enter into the medical college, you will be studying other nuclei also associated with the limbic system. Now, so that completes with the brain. Now, next part of the CNS, that is the spinal cord. So next one, spinal cord. Now spinal cord, you see, is present inside the vertebral column. You know that it is protected inside the vertebral column, and it extends from the medulla, we have just now mentioned. So just from the upper surface of the first cervical, and if you see the spinal cord, it is broader in the beginning and then it narrows down, and then once again it protrudes and then finally tapers. Okay, like this. And then there is a non-nervous part going like this. So in this case, the level here, from where it is starting, so that is the level of your first cervical, C1. That's why I'm writing. And same way here, where is it ending? That is at second lumbar. Now some books write it first lumbar, some write it 2, but it is between lumbar 1 and 2, doesn't matter. Okay, not so close choice you are ever going to get. So from C1, upper surface of C1, that is our first cervical, that was atlas, right? From atlas to the second lumbar. And after that, this part that you see here, this is phylum terminale. Phylum terminale. This part is non-nervous. No neurons are here. It is just the meninges. This would be extending up till the coccygeal vertebra, first coccygeal, right? And this part here, this is called the conus medullaris. Conus medullaris. And from this you know very well that the spinal nerves are arising. I'm not making all of them, just to give you an idea. And these are, of course, this is spinal nerve, which we have mentioned. These are 31 pairs, and all of them are mixed. Mixed means which have both sensory and motor fibers, that is afferent and efferent fibers. So all of them are mixed, all 31 pairs. So easy to remember. And now if we take a section TS through the spinal cord, what we will see is, so let us make it here. This part is over, I suppose. by this time. So in this, what you will find is that in the section, it will appear like this. There is a central CSF filled cavity, and just like any CSF filled space, it is also lined by ependyma. Now inside here, there is the grey matter. I told you the grey matter becomes internal in the medulla, and the same thing continues in the spinal cord. So this is all grey matter here. This is all grey matter, okay? And now on one side I am showing the same thing on the other side, but anyway we will take it on one. And then it goes like this, okay? So here let us label it. This is the central canal, or the neurocele, as I told you, central canal. Here in the center I can write here, this is all grey matter, and this all here is white matter, okay? Now you will see the grey matter here also is projecting into the white matter, and these projections are called horns. So here this one is dorsal horn, dorsal horn. This one is lateral horn. This is ventral horn, okay? Similarly, the white areas which are in between the grey areas, that is called funiculus. So here also, these are funiculus. So I'm just writing at one end. So you will know when funiculus is given in the question, we are talking about white areas, horn are the grey areas, okay? Then this structure that you see here is, this is a ganglia, let me mark, dorsal root ganglia, ganglion singular, okay? This is dorsal root. This one is ventral root. Now you see this, let me put here, I hope everybody is able to see. Here this is the cell body, and this goes like this. And inside there are a number of neurons, and from here it is synapsing with another, and this goes like this. This is just one fiber I have drawn, but in the root, of course, number of fibers would be there. So this is just for example's sake. So you see in the dorsal root, all the fibers which are present, all fibers, they are all of them sensory fibers, that is afferent fibers. So here these are afferent fibers, or the sensory fibers. So the dorsal root has only afferent fibers, and the ventral root has the efferent or the motor fibers. Now you see when these two join together, the ventral root and the dorsal root join, this becomes the spinal nerve. Now this is the spinal nerve, you see, and in the spinal nerve you are finding both the fibers, the sensory and the motor fiber, that makes it mixed, okay? One more thing, in the dorsal root ganglia, this example we had done I think in the tissues, the type of neuron that you see here, that is what pseudo-unipolar neuron. So in the pseudo-unipolar neuron, I told you that the cell body is at one end, only one process is coming out, and that process is then dividing, it is bifurcating, so that one is behaving as the dendrite and the other is axon. So you see, since it is a sensory fiber, so the sensory fiber would be ending or starting rather I should say from a receptor. This is starting from a receptor, the direction of the impulse will be this. This will be the direction of the impulse. And this motor fiber will be carrying command from the spinal cord to the outside, and this is ending at the effector. An effector, as I told you, can be a muscle, it could be a gland, right? So this is how the entire pathway is there. So you have all these structures, and one more let me mention here. This one is called the interneuron, or also called relay neuron. Interneuron or the relay neuron. And just for keeping things simple, I have drawn only one, but there would be many relay neurons. So relay neurons are those neurons which are entirely inside the CNS. Okay, so their fibers are not going out of the CNS, they are totally inside. Okay, so in this manner you see the TS of the spinal cord. So you can once take down the diagram, and I think that in the diagram itself everything is there. Then we can talk about reflexes. Done? So at least I can take off this part here. Now next thing you see, reflex action. Now reflex actions, you see, this is a kind of response, and it's a very quick response. That is our body's mechanism for protection, for defense. And so this is a involuntary response for a particular stimulus which occurs on any peripheral part of the body. So this is a spontaneous involuntary response against a stimulus. Now the entire pathway for reflex action, that is called the reflex arc. Reflex arc. So this is the neural pathway of reflex action, and in this neural pathway you will find there are 5 components. Starting from here, it's already drawn here, you see. First one would be the receptor. So from the receptor receives the stimulus, impulse goes from here via afferent nerve. So afferent nerve, this would be our second component. Then it comes into the CNS. So I am saying CNS, it could be spinal cord, it could be brain, right? Because you see, even vomiting, etc., we had used the term reflex, vomiting reflex, right? So those are, as I was telling you, they are certain reflexes which are already wired into our system. So this is the CNS is the third one. Then efferent or the motor nerve, this is the fourth. And finally the action will come on the effector, that is the fifth. So these are the 5 components which are making up the reflex arc, you see. Okay, so this is already drawn over here, and you can easily, if you want, you can write it also. Okay, so this is one. Now coming to some types of reflexes, because that would be more important for you. Just a sec. So in your NCERT, like they have given example of a knee-jerk reflex. Now knee-jerk reflex, you see, is a monosynaptic reflex. Monosynaptic reflex. Now when we say monosynaptic reflex, that means there is single synapse in the entire reflex arc. And how is that made possible? Because there are certain sensory fibers which will be directly synapsing with the motor neuron here. So that means they are bypassing the interneuron. So you see only one synapse here, nothing else. So such synapses are, because I told you the more are the number of synapses, it will take more time. But since number of synapses is just one, so this would be a very, very quick response, right? So monosynaptic reflex, in this case we can mention the sensory fiber directly synapses with motor fiber. Okay, that is monosynaptic. And the example here is given as knee-jerk reflex. So in knee-jerk reflex, this is basically the doctors carry out this kind of, it's kind of, you can say, a test. If a person has got some injury in the spinal cord, because this is a spinal reflex you see, and to check it whether the nerves are functioning properly, they will carry out this test. So in this case, the person is asked to sit on a chair, okay, and so that the leg is bent at right angle here, okay. So here the leg is bent and it is on the ground, okay. Now the doctor will strike here just beneath the kneecap by a hammer, of course rubber hammer, right? So the doctor will strike here, and as soon as it is struck, the leg would extend. It will be moving, it will kick out literally, okay? So it will move like this. So here you will see that at the base of the kneecap, here the receptor is present. From that receptor, the impulse is going to the spinal cord, and from there the motor command, and here you see we have at the back of the thigh we have the hamstring. Hamstring muscle is going to contract and the leg stretches out. Okay. And in the NCERT, however, you will see there are two efferent pathways they have made. Two efferent pathways, and that is because of that students have a lot of confusion that why two efferent pathways are there. Now you see our body never depends on one thing. There is an alternate also. Okay, the major reflex, if you get a question what kind of reflex is knee-jerk reflex, you will write monosynaptic, no problem there. But there is one more pathway which is certainly through the interneuron, it is involving the interneuron, but that one is coming to this quadriceps here. This is coming to the quadriceps. The quadriceps is on the upside of the thigh, and it is the quadriceps which is keeping the leg bent. This is okay. So that other pathway, what it is doing is it is inhibiting the further contraction of this quadriceps. So that because if the quadriceps it remains contracted, the leg will remain in the bent position. So that is what it is doing. Okay, so that is the alternate part. Needn't bother about that. The main thing that you have to learn is that this knee-jerk reflex is very much a monosynaptic reflex. Okay. Now the thing is that say there are certain reflexes where not one but many interneurons will be used in the pathway, and those are called the polysynaptic reflexes. Okay, so let me mention the other one. So I think I can this diagram I can remove. So next one, polysynaptic reflexes, and these are more common. More of the reflexes are of this variety, polysynaptic reflex. So in this case, number of interneurons are involved in the reflex arc. Okay, so number of interneurons are involved in the reflex arc. Okay, and the example of such polysynaptic reflex is the withdrawal reflexes. Now withdrawal reflexes is when a person moves away from a stimulus, withdraws itself from the stimulus. Like the usual example you are given, touching a hot plate. Okay, like removing the hand on touching hot plate. Now in this case you will see that there are a number of things which will be occurring simultaneously. So as soon as a person has touched the hot plate, they immediately remove the hand, and with a jerk, of course, even the body also moves, because that means different motor commands are going, you see. And then with the other hand they'll start rubbing it, they'll be blowing over it, and some expletives will be coming out from the mouth, and so many changes are occurring at the same time. So many motor effects can be seen. So that means number of interneurons are involved, and so many different synapses to the motor nerves are involved at the simultaneously manner. Okay, so this is what is the withdrawal reflex. Fine. Then coming to one more criteria of reflexes, that is simple one, although that is on the basis when this reflex develops, on the basis of development. So there are two types, we can say, one which we are born with. Okay, so on the basis of development, reflexes are of two types. Okay, one is inborn, or which are unconditioned reflexes, also the term is used, unconditioned reflexes. So inborn, of course, you know, by birth. So there has never been a condition created. This is already present. The entire pathway is there in our system. Like for example, salivation. When we put food in the mouth, the saliva secretion starts. So all those reflexes, you see, they are very much of this inborn variety. And for a mammalian baby, you see, mammalian baby, nobody teaches them to suckle milk. But as soon as the baby is born and they are brought near to the nipples, they'll start suckling the milk. So suckling of milk, salivation on salivation, these all are inborn reflexes. Then another one is conditioned reflexes, and this one, you know, when the mind is conditioned to a certain thing. Okay, and these are the famous, after the famous experiment by Pavlov, Ivan Pavlov, the dog and the bell experiment. I hope everybody knows that he trained his dog in such a manner that when he used to ring the bell, just immediately after that, he used to present that dog with food in the form of meat powder, and he had placed certain apparatus also, like a tube from the mouth, so that he could measure the saliva and everything. So slowly the dog got conditioned that ringing of the bell means that food is going to be presented. And then after some days, once the conditioning was over, he just rang the bell but didn't give the food, but still salivation occurred. So that is how, so this one is conditioned reflex. So this one is it is learned. This reflex is, we are not born with that reflex, but we learned it because of repeated actions. Okay, so this is learned by repeated actions, and or we can also, in other words, we can say we associate it with certain condition, or by associating it with certain condition. Okay, so like, of course, conditioned reflexes, you can talk about like cycling, moving your legs on the pedal. You needn't look it in the beginning when you are learning how to ride a bicycle. Certainly every time you are focusing sometimes on the handle, sometimes on the pedal, but once you learn it, you needn't look even. Okay, so cycling, playing any musical instrument, playing a musical instrument, typing, so on, right? So this much we'll do about reflexes. It's quite comprehensive. You won't get question about beyond that. And sometimes you see you do get about salivation is somewhere where we give both the examples. Like there is a foods you had never eaten it. Like just for example, let us say some Mexican cuisine it is, and you had never ever tasted it. So while you will not salivate looking at that food. Now once you do eat it and you relish the taste, then from the next time onwards your salivation for that food also will become. But there of course that would be a learned reflex. That would be learned reflex. That is not just the food, because naturally you didn't know about that food. Okay, so you'll see in the question accordingly, for a new type of cuisine if it is mentioned, then it will become a conditioned one. Now after this we can move on to a little about PNS, because PNS as such in detail is not there in our NCERT, but we will be doing ANS anyways. Autonomic nervous system we have to do. Okay, so PNS, that is peripheral nervous system, and peripheral nervous system, as we mentioned, this comprises two, that is cranial nerves and spinal nerves. Now cranial nerves, all 12 pairs. I don't know whether you have learned it or not, but let me tell you, not all are important, but some of them certainly, because which we will relate with the receptors, those certainly we will be learning, right? So all cranial nerves, you needn't think about learning them, but the major ones which are associated with our main sensory organs, like for olfaction, gustation, so on, photoreception, those all we will be doing. So I am not doing it presently, because with the sensory structures you will relate them. Spinal nerves, we have already just now mentioned that these are 31 pairs. I can give a formula here, of course. So in which area how many are there? So we can write it like this: C8, just like we had the vertebral formula, but there it was cervical was 7, but spinal cervical nerves are 8. So C8, T12, L5, S5 and coccygeal 1. So if you add on these numbers, you will see it will be 31, right? So this is how we can write this formula. Now, one part of the peripheral nervous system, you know, is autonomic nervous system, ANS, autonomic nervous system. Now you see what is so special about autonomic nervous system. So at least let me first give you the idea about it, and then the later part of it, that is the difference between sympathetic and parasympathetic, that we can do in the next class. Okay, so here let us put like this. Suppose this is our central nervous system, and since we are going to talk about motor nerves, so in a motor nerve, the cell body, the cyton, is always going to be inside the CNS. You had just now seen in the motor fiber, the cell body was inside the gray matter, isn't it? So here also the cell body is always going to be inside the CNS. In autonomic nervous system, you see what happens is that this doesn't reach up till the effector, rather it ends beforehand. And this will be ending here in a ganglia. There is a ganglia here, and here it is synapsing with other neurons, and from there the impulse is carried to the effector. Okay, so in this case, you see this fiber can be called preganglionic fiber. This is the autonomic ganglia. Autonomic ganglion. This would be postganglionic fiber. Okay, and this is, since we are talking about autonomic, so of course I can make a smooth muscle or a cardiac muscle. So here the effector is going to be involuntary muscle, and when I write involuntary, so we have both smooth as well as cardiac, and also the glands, these are affected. Okay, So in this case, what you see is that, first of all, what is the neurotransmitter released here? So preganglionic fiber is going to release acetylcholine. And this one I have drawn with a different color because this is releasing some other, that is norepinephrine. And I told you in the last class that in the peripheral nervous system only two neurotransmitters are used, and you can see both of them. So if this is the arrangement, this would be our sympathetic nervous system. So you know that ANS has two divisions. One is sympathetic, the other one is parasympathetic. So let us see how the parasympathetic is arranged. So in the parasympathetic, you will see that once again the neuron is here, and this is also ending in a ganglia. Here it is synapsing with another neuron, and from here the impulse goes on to the effector. Okay, so here you see this is the preganglionic fiber, this is the postganglionic fiber, this is the autonomic ganglia. But what is the difference? Here both of them release acetylcholine. Okay, both pre and post synaptic fiber, they release acetylcholine. And this is also ending on the same thing, that is involuntary muscles and glands. So you see the effect of both of these, that is sympathetic and parasympathetic, that would be almost antagonistic. If one of it is increasing a certain activity, the other one is decreasing it, and vice versa. And this is what maintains homeostasis. Now homeostasis is our internal environment. So the internal environment is maintained in a steady state because of this autonomic nervous system. Okay? So autonomic nervous system, one thing you can always remember is that autonomic nervous system is a two neuron system. So here from the CNS, the neuron, no fiber directly reaches up till the effector. Rather it first synapses with some other neuron, and so you see in the entire pathway we are using two neurons I have mentioned. So it's a two neuron system. And if you will see the somatic one, the other day we mentioned that PNS has two parts. That PNS is having, one is the somatic nervous system and the other is the autonomic nervous system, isn't it? These two parts. Now this somatic nervous system will be here also, if I'll just mention here so that you have the comparison with you. So here if I put CNS, so here this is the motor neuron, and this motor neuron is directly reaching up till the skeletal muscle, you see. So here it is the skeletal muscle, and here what is being released? Acetylcholine, and this is our somatic nervous system. So you see somatic nervous system is a one neuron system, and this neuron, as I said, this is motor neuron. Here also these all are motor neurons. Okay. So in this case, as this is how the arrangement is, at least the basic arrangement, you can see that what kind of arrangement we can see here. Let me see if there is any question. Okay. Right. So once you are through with this, the basic difference, then we can talk about that which part of the PNS, that which nerves are contributing to sympathetic and which one are contributing to parasympathetic. Okay. So next we can take a little part of it. Up till here I think it should be over. Okay. So you see, starting with these, first or simply I will put here sympathetic nervous system. Now in sympathetic nervous system, this is, you see, also called thoracolumbar outflow. Why so? Because the nerves which are contributing to it, they are the thoracic spinal nerves and the lumbar spinal nerves. So this is formed by thoracic and lumbar spinal nerves. Okay, and which thoracic? Thoracic, you see, are 12 of them, so all 12, T1 to 12, all of them. And lumbar are 5, but out of that only 1 to 3. So that means these 15 pairs of spinal nerves, you see, they are making our sympathetic nervous system. And in this sympathetic nervous system, you have seen that the preganglionic fibers, now whatever they release, that's how the name is given. Preganglionic fibers are cholinergic, that means they release acetylcholine. Preganglionic fibers, these are cholinergic, and postganglionic fibers, are noradrenergic. Noradrenergic because they are releasing as this one. Okay. Then another thing that is sympathetic nervous system is called, this is responsible for the what is that, fight or fright response. Okay, so that means in emergency, this is going to work in emergency. So what all it does during emergency, let us see that. So one is that it prepares the body for emergency. So in that case, what is happening? Pupils dilate. Pupils dilate, that is, more light will fall on the retina and you can properly see your enemy or the way to escape, right? Pupils, they are dilating. Then there is piloerection. Piloerection, that is the body hair, you see, because you see the hair, it is normally bent at an angle, and you know that the base of the hair, there is a hair follicle, and to that hair follicle there is a muscle attached, that is called the erector pili muscle. So the contraction of the erector pili muscle, because of this the hair is going to be raised. The hair is raised, that's what is piloerection. And even in cold, when we are in a colder climate, then also it happens, because air gets trapped in this the space created, and air is a good insulator, so that way we are protected from cold. Even then also it happens. Okay, so piloerection, so this one, there is contraction of erector pili muscle, contraction of erector pili muscle, and hair is raised. Okay, so this is there. Then some of the things you already know, that heart rate increases, breathing rate increases. So there is increase in heart rate, breathing rate. There is increase in blood glucose, because we would require energy to run or fight. Just a sec. Okay, so there haven't been any doubts, I think, for some time. Okay, so increase in blood glucose, increase in the blood glucose, and this is by glycogenolysis in the liver. So in the liver there is breakdown of the glycogen, so that glucose is released into the blood. Okay, then it's almost time, but anyway, one more function we'll mention. When you are frightened and you are running, then of course at that time the digestion takes a back seat, right? So at this time peristalsis and all the gastrointestinal secretions including salivation, all that are inhibited. So peristalsis and gastrointestinal secretions are inhibited. Okay, this is there. So some more functions we'll list out, I think, in the next class. It's almost time, and so we can wind up today at the moment. Yes, only 1 minute left. So thank you students anyways, and up till here just go through it. There have been lot of part which we have covered today. So do read the NCERT and keep the class notes with you so that you have a complete picture, right? So that's all for now.